Are your peppers struggling to reach their full potential? What if you could unlock explosive growth and massive yields, all without relying on synthetic fertilizers? We're about to show you how we're building a groundbreaking microbe farm specifically designed to supercharge your capsaicin plants, creating a living soil ecosystem that practically grows itself. The secret to peppers that produce abundantly while developing exceptional flavor and heat lies not in bottles or bags, but in cultivating the precise microbial communities that these plants evolved alongside for millennia. The transformation from struggling pepper plants to vigorous, heavily producing specimens happens not through the visible interventions we make above ground, but through the invisible biological networks functioning in the root zone, where trillions of microorganisms work in concert to support plant health. Every teaspoon of healthy soil contains more microbial organisms than there are humans on Earth. Bacteria, fungi, protozoa, and nematodes form these really complex food webs that break down organic matter, cycle nutrients, suppress diseases, and even directly communicate with plant roots through chemical signaling. Peppers, like all plants, didn't evolve to extract nutrition directly from soil minerals. Instead, they developed symbiotic relationships with specific microbial partners that provide these services in exchange for the carbon-rich exudates plants release through their roots. Understanding this fundamental truth, it really shifts our entire approach from just adding nutrients directly to actually cultivating the biological communities that make nutrients available naturally and continuously. Generic potting mixes and even quality garden soil, well, they lack the specialized microbial populations that capsaicin-producing plants specifically benefit from. Instead, they contain a random assortment of whatever organisms just happen to colonize during the soil's formation or production. Peppers in their native Central and South American habitats grow in soil ecosystems dominated by particular fungal and bacterial species that have co-evolved with capsicum species over millions of years, developing mutually beneficial relationships that maximize plant vigor while the microbes secure reliable carbon sources. When we grow peppers in soil lacking these specialized partners, plants must work harder to access nutrition, show reduced stress tolerance, and often fail to develop the complex flavor compounds and capsaicin concentrations that characterize exceptional fruit. The solution isn't adding more fertilizer to compensate for poor microbial support, but rather engineering the biological environment that allows peppers to thrive through their natural partnerships. Sourcing beneficial microbes for your pepper-specific farm begins with identifying the most vigorous, disease-resistant pepper plants in your garden or local growing community. You know, their success almost certainly reflects superior microbial partnerships in their root zones. The rhizosphere soil adhering directly to healthy pepper roots contains concentrated populations of the beneficial organisms we want to propagate. Plants actively recruit and feed their preferred microbial partners through selective root exudation. Carefully excavate soil from around the roots of champion pepper plants, taking samples from the zone within 2 inches of active root tips, where microbial density and diversity really peak. This rhizosphere soil serves as your starter culture, containing not just individual species, but entire functional communities that work synergistically to support pepper health in ways isolated strains simply cannot replicate. Creating a simple microbial nursery for propagating these beneficial organisms requires nothing more than non-chlorinated water, unsulfured molasses or other simple sugars, and adequate aeration to support the explosive population growth these conditions trigger. Combine 1 part rhizosphere soil with 10 parts water in a bucket, add a tablespoon of molasses per gallon, and provide continuous aeration using an aquarium pump and airstone for 24 to 48 hours. This actively aerated compost tea process multiplies beneficial bacteria populations by factors of thousands, while fungal populations increase substantially as well, creating a living inoculant teeming with the pepper-supporting microbes extracted from your source soil. So, the molasses actually feeds bacterial growth preferentially, while aeration prevents those anaerobic conditions that would let pathogenic organisms take over. This way, your finished tea ends up containing mostly beneficial species. Scaling up production to treat whole pepper beds means you'll either need to make larger batches of compost tea, or, set up semi-permanent microbial farming systems. That could be worm bins or compost piles that you manage specifically to favor the organisms that are really good for peppers. Vermicompost, which you get by feeding composting worms a diet rich in pepper plant materials, naturally ends up concentrating the microbial species linked to capsaicin producers. These organisms actually colonize the plant matter, and then survive the trip through the worm's digestive system so they can populate the finished castings. Similarly, compost piles built with significant proportions of pepper foliage, roots and fruit waste become dominated by pepper-associated microbes, while the thermophilic composting process eliminates potential pathogens. These approaches create ongoing sources of pepper-optimized microbial inoculants that can be applied throughout the growing season without repeated trips to source Rhizosphere soil from champion plants. The substrate architecture supporting your microbial community matters as much as the organisms themselves, because even the most beneficial microbes cannot function in soil lacking the physical structure and chemical conditions they require for survival and activity. So, pepper-supporting microbes really thrive best in soil where the pH sits between 6 and 7. You want enough nitrogen, but definitely not too much, and abundant organic matter is key. It gives microbes both food and a place to live. Plus, having excellent drainage is crucial, since it prevents those soggy, anaerobic conditions that would favor pathogens instead of the beneficial organisms we're after. Building this ideal environment, well, it actually starts by mixing in substantial quantities of mature compost into your beds before you plant. The goal is to get your organic matter content up to at least 5%, but honestly, it's even better if you can reach 8 or 10%. That's where you'll see optimal microbial activity. The compost does double duty. It provides an immediate boost of microbes and also serves as a stable food source, sustaining those populations all through the growing season as the fresh organic matter slowly breaks down. Mineral amendments, including things like rock dust and biochar, create additional benefits that specifically support pepper-favorable microbial communities, while also improving overall soil function. Rock dust provides slow-release mineral nutrition that reduces dependence on soluble fertilizers, which, can disrupt microbial populations through sudden nutrient flushes and salt concentrations. At the same time, it supports bacterial species that are specialized in weathering mineral particles to access nutrients. Biochar acts as a kind of permanent habitat for microbial colonization. With its massive surface area and complex pore structure, it provides protected spaces where organisms can establish stable communities, sheltered from predation and environmental fluctuations. Introducing your custom microbial inoculant at transplanting time gives pepper seedlings immediate access to beneficial partnerships during the critical establishment phase, when root systems are most vulnerable to pathogen attack. Drench the transplant holes with actively aerated compost tea before setting seedlings, ensuring root systems contact the microbial inoculant immediately rather than spending days or weeks searching for beneficial partners. Follow-up applications every 2 to 4 weeks throughout early growth maintain robust microbial populations as root systems expand into new soil zones, with application timing ideally synchronized to periods of active root growth when plants are most receptive to forming new symbiotic connections. Monitoring microbial community health involves, observing both plant vigor indicators and direct soil assessments, like the presence of white fungal mycelium that you might spot when you gently pull back mulch layers or dig small exploratory holes near your plants. Disease suppression through competitive exclusion and direct antagonism represents, honestly, one of the most valuable services robust microbial communities provide. This can potentially eliminate the need for any fungicide applications, even when environmental conditions really favor pathogen development. Beneficial bacteria and fungi colonize root surfaces in the surrounding soil so densely that pathogenic organisms just can't find purchase to establish infections. It's basically armoring plants against disease through sheer population dominance. Many beneficial species also produce antibiotics or other compounds that directly inhibit pathogen growth, creating chemical barriers that supplement the physical protection of competitive exclusion. Mycorrhizal fungi, particularly arbuscular mycorrhizal species that colonize pepper roots extensively, improve plant immune function through systemic signaling that primes defense responses throughout the plant, making tissues more resistant to both disease and insect attack. Water management becomes dramatically easier when soil supports thriving microbial populations that improve structure and create organic coatings on soil particles that enhance moisture retention. Fungal hyphae physically bind soil particles together while secreting sticky compounds like glomalin that stabilize aggregates against erosion and compaction. This creates the porous structure that allows soil to absorb water rapidly during irrigation or rainfall, yet drain freely to prevent waterlogging. The organic matter that feeds microbial communities also functions as a sponge, holding several times its weight in water and, releasing moisture gradually as plants need it. This reduces irrigation frequency while preventing the drought stress that limits pepper production. Peppers grown in biologically optimized soil routinely require 30 to 50% less irrigation than those in conventional substrates, with better drought tolerance when water becomes limited. The connection between soil biology and fruit quality surprises many gardeners, but actually, extensive research confirms that peppers grown in microbe-rich living soil develop more complex flavor profiles and higher capsaicin concentrations than those produced with synthetic fertilizers. Mycorrhizal associations, interestingly enough, enhance plants' ability to access micronutrients, including sulfur and various trace elements that serve as precursors for the aromatic compounds contributing to pepper flavor complexity. The balanced gradual nutrient availability created by microbial nutrient cycling prevents the excessive vegetative growth that high-nitrogen synthetic fertilizers trigger, which in turn allows plants to allocate more energy toward fruit production and the synthesis of secondary metabolites, including capsaicinoids. Taste tests consistently demonstrate that peppers from living soil systems possess more nuanced flavors, better texture, and, in the case of hot varieties, more pleasant heat characteristics with less bitterness than conventionally grown comparisons. Adapting the microbe farming approach to plants beyond peppers simply requires sourcing your starter cultures from Rhizosphere soil around vigorous examples of whatever species you want to optimize for recognizing that each plant type recruits somewhat different microbial communities. Tomatoes, which are close relatives of peppers, benefit from very similar organisms and can often use the same microbial inoculants successfully, while brassicas prefer different bacterial populations and squash family plants associate with distinct fungal partners. Building separate compost tea batches from Rhizosphere samples of each major plant family you grow allows you to tailor applications to specific crops. Though universal inoculants made from diverse compost still provide substantial benefits when crop-specific versions aren't available. Troubleshooting. Struggling microbial populations typically reveals that environmental conditions rather than the organisms themselves are limiting establishment. The most common culprits include chlorinated water that kills bacteria, excessive synthetic fertilizer applications that disrupt biological processes, or soil pH outside the range where beneficial organisms thrive. Switching to rainwater or allowing tap water to sit for 24 hours before use eliminates chlorine issues, and by reducing or eliminating synthetic inputs, microbial populations can recover and re-establish those functional communities. Testing and adjusting soil pH into the optimal range for your target crops addresses perhaps the most fundamental requirement for microbial success, as even perfectly cultured organisms cannot function in extremely acidic or alkaline conditions. The future of sustainable gardening and agriculture increasingly points toward biological management approaches that work with natural processes rather than attempting to override them through chemical inputs. Every garden that transitions from synthetic dependence to biological support demonstrates that superior results emerge when we focus on creating conditions where beneficial organisms thrive, then allow these microscopic partners to handle the complex work of nutrition, disease management, and stress mitigation. The initial investment in building microbial farming capacity really pays dividends that just keep compounding over the seasons. The soil keeps improving continuously, and input requirements actually start to decline as the biological function becomes more and more self-sustaining. Your journey into microbe farming begins with understanding that soil health isn't measured in NPK numbers, but in the diversity and activity of the organisms living within it. We encourage you to experiment with these techniques on even a small scale, maybe starting with just a few pepper plants treated with custom microbial inoculant while leaving others as controls, so you can observe the differences firsthand. Subscribe to the Living Soil Lab for more deep explorations of soil biology, practical techniques for building living soil systems, and ongoing documentation of methods that transform gardening from chemical dependence to biological partnership. Together, we're pioneering a gardening revolution that recognizes microscopic organisms as the true foundation of plant health and productivity.